Today I'm going to discuss endocrine histology, and we're going to look at just three tissues. We're going to look at the thyroid gland, the suprarenal, and the pancreas. Now let's begin by looking at the thyroid gland. And when you look at the thyroid gland, the first thing you notice are round structures which are known as follicles, and inside these follicles you have a pink homogeneous material which is called the colloid. Inside colloid, which is made of iodinated thyroglobulin, you find the two thyroid hormones, T3 and T4. Calcitonin, which is another hormone produced by the thyroid gland, does not make its way into the colloid. It passes out of the C cells directly into the bloodstream. Now these follicles are lined by cuboidal epithelium, and you can detect that it's cuboidal epithelium when you see that the cells are as tall as they are broad. The blue arrow that you notice points to C cells, and these are the cells which produce calcitonin, and as I mentioned earlier, calcitonin is one of the thyroid hormones, but this is not present inside the colloid. Let's move on to the next slide, which is that of the pancreas. Now the pancreas has both an exocrine and an endocrine portion to it. The exocrine portion is concerned with digestion, so we won't be discussing that now. We'll only look at the endocrine portion, and in the endocrine portion you notice these circumscribed areas which are known as the islets of Langerhans, and these contain your alpha and beta cells which produce glucagon and insulin. Now while both these images are pretty distinct and clear, and you can see the islets circumscribed as tiny islands, sometimes in the lab when you look at slides you may not always see them so distinctly marked out. They may just appear as tiny cells in almost like, flowing like a stream, and that just varies from tissue to tissue. Next to the islets you will notice the other structures which belong to the digestive part of the pancreas, and these are known as serous acini. These are again round structures, darkly stained, and they produce enzymes and have nothing to do with the endocrine part of the pancreas. Let's move on to the suprarenal gland, and this gland is different in that it produces totally different types of hormones, and it has two very distinct areas. We have the cortex and we have the medulla. Now in the cortex, the way you identify the cortex is by first looking for the capsule, and this consists of connective tissue, and you can see those collagen fibers up there. And right next to the capsule, the first layer you see is the zona glomerulosa. And the zona glomerulosa consists of cells which arrange themselves in a ball-like fashion, much like the glomerulus of the kidney. And this zone produces mineralocorticoids, of which aldosterone is the main one that you learn about. Next to the zona glomerulosa, you see the next layer, which is where the cells are arranged in long bundles, and this is called zona fasciculata. And this zone produces glucocorticoids, of which the main glucocorticoid that you know of is cortisol. Further down, next to the zona fasciculata, is the zona reticularis, where all the cells are connected in a network-like fashion, and this zone produces androgens. So in order to really identify these zones, you must first look at the capsule and go from there. Next to the zona reticularis, what you see is the medulla, which is darker staining, and ideally it should have large cells which are the sympathetic ganglion cells, because this medulla is connected to the sympathetic nervous system. Unfortunately, in both these images you do not see those ganglion cells, but you can definitely make out the color difference between the cortex and the medulla, and this area is darkly stained. Now with that, we wrap up the endocrine histology. This is a short tutorial, And I would suggest, like always, that you rewind and look at these images fairly often so that you're comfortable being able to identify all these different tissues. And with that, I'd like to wish you all a really happy day.